A team of scientists from the Life and Medical Science Institute of the University of Bonn and Caesar Research Center are hot on the trail of cardiac valvular defects and have made important progress. Professor Michael Hoch from the University of Bonn explains why this step is so important. Cardiac valvular defects are unfortunately the #1 cause of death among newborns, and this is why it is so important to understand how these defects occur. For this reason, LIMES and Caesar have worked together to future research the so-called CRELT1 gene. Another special thing about this project is that this CRELT1 gene was identified as a risk gene in human beings some years ago, and more specifically as a risk gene for cardiac valvular and heart wall defects. In this way, by examining the CRELT1 function in mice, we have identified a significant molecular signal path that leads to the formation of the valves of the heart, and which, when it is defective, can result in failure of the valves of the heart. The team made use of the mouse model for new findings concerning the signal path. Responsible for this, among others, Dr. Elvira Maas. She explains the approach.
Because it wasn't exactly known what causes this gene or protein in the heart, we approached the task and deactivated the gene in the mouse. This is done with a so-called knockout. That means one removes the gene from the genome and then observes what happens with the mouse. What we saw is that the gene is in fact important for the development of the heart, and that the mice or the embryos without this gene, meaning in which it has been deactivated, die from a heart defect. At this point, we had the idea of cooperating with Dagmar Wachten, who has expertise in this field.
Dr. Dagmar Wachten from the Caesar Research Center is an expert in the subject of stages of development in the heart. Because this is similar in humans and mice, the new findings from the mouse model are important for human medicine.
The mouse is an ideal model system for heart development in humans. The development period of the mouse is relatively short, meaning 19.5 days. Human development takes considerably longer, but the good thing is that mouse and human development are nearly identical with reference to heart development. This means that one can carry over the short periods of time for the mouse almost identically to the heart development of people. We are thus convinced that the findings gained from the mouse can be carried over nearly 100% to humans, because the signal paths are very similar. The reverse conclusions at the molecular level can be directly transferred to humans.
This is in turn very important for medicine. Professor Hoch emphasizes that it will take some time until the findings are applied, and yet our findings will in any case lead us better understanding these embryonic heart disorders, and to our ultimately being able to develop new therapy approaches in future by clarifying the molecular causes. In addition to this, the research results will have an impact on the treatment and diagnosis of heart defects in adulthood and on transplantation medicine.